So this is an animal cell model, and let's take a look at some of the cell structures we've got here. Out here, we've got the cell membrane, that phospholipid bilayer that controls what goes into and out of the cell. It would contain the glycocalyx that helps to identify this cell to other cells, enzymes, transport proteins. It's kind of like a security guard for your cell. These two dots, the yellow and the white here, are either peroxisomes or lysosomes. Both of those are organelles that will help dispose of debris or worn out parts, or if this cell had endocytized a pathogen or, say, a bacterium, those would have digestive enzymes that help to break down whatever you need broken down in your cell. Here we see a mitochondrion, and here are a few more. Collectively, they're called mitochondria. They're the site of aerobic cell respiration. So we're using glucose and oxygen to extract as much energy as we can and make ATP. This is the nucleus. And the function of your nucleus is to store and contain your genetic material, your DNA. This is a nucleolus. That is where we're making our RNA, which is then going to leave the nucleus through these nuclear pores, and then it becomes part of the physical structure of a ribosome. So we're a little bit closer here, and now you can see there's some thread-like structures here throughout the nucleus. Those are chromatin. So those are your chromosomes, but in an uncondensed form, because we would assume that this cell is not actively dividing. This is rough endoplasmic reticulum. The primary jobs of our rough ER or rough endoplasmic reticulum are to synthesize and then modify proteins that are either going to become part of the cell membrane, or they are going to leave the cell through exocytosis and go to another part of the body. Over here, we have smooth ER, or smooth endoplasmic reticulum. It's called smooth because if you look at it versus rough ER, you can see that the rough ER has ribosomes, and there's also some ribosomes down here in the cytoplasm. The ribosomes are a site for protein synthesis, which is why we've talked about what rough ER does. Smooth ER has many functions, but here's a few. So one thing they do as a specialized structure called the sarcoplasmic reticulum in muscle cells is that they store calcium that's needed for muscle contraction. Smooth ER, it also participates in detoxifying the cells, so they'd work with lysosomes and peroxisomes. You'd see a lot of those in, say, the liver, whose one of its jobs is to detoxify blood or to strip waste products out of blood, or toxins or drug metabolites out of blood. Smooth ER is also really important in synthesizing lipids for the cells, such as phospholipids that make up the cell membrane. So next, let's look at the Golgi body, here and here. Sometimes it's called Golgi body or Golgi apparatus or Golgi complex, depending on the text that you're reading. So if you look at smooth ER and the Golgi body, they look fairly similar, especially if you look over here, other than color. And one way that you can tell the difference is that smooth ER tends to be closer to the nucleus, and the Golgi body will usually in a model show these vesicles that have come off of it. And those vesicles are packaged materials that were modified by the Golgi body for export out of the cell or for transport around the cell. Sometimes I kind of think of the vesicles as sort of like a Ziploc bag or a storage bag that's, say, in your refrigerator or in a closet. These are the centrioles, and the centrioles become pretty important when we perform cell division because they're going to make spindle fibers that attach to the sister chromatids and then help pull those chromatids apart and to either side of a new and developing cell. These structures are microvilli. So those are fan foldings of the cell membrane, and they become especially important throughout the digestive system and in some places of the kidneys where there's a considerable amount of absorption or reabsorption and secretion of products. Fan folding the cell membrane like this increases the amount of surface area for absorption or secretion. And then down here, these lines that you see, and then you can also see a few in the cell here. Those are parts of the cytoskeleton. Now we can't tell specifically whether they're microtubules or microfilaments or intermediate filaments, but we can tell that they're part of the cytoskeleton, that sometimes they serve as tracks that help to move products across a cell. Other times they'll help to anchor cells to each other, say in the epidermis of the skin. And then lastly, this bluish background that you see throughout the cell in which the organelles are embedded is cytoplasm or cytosol. So the difference between those two terms are that cytosol is that gel-like watery matrix in which the organelles and the nucleus are embedded. It's about 90% water. So when you're dehydrated, one of the things that happens is your body pulls some of that water from the cytosol to the bloodstream to help you maintain normal blood volume. Cytoplasm is the cytosol plus the nucleus plus all the organelles and chemicals that are in that cytosol.